Go ahead and hit record. Okay? So we are doing cellular respiration. Say your cellular respiration chapter. Everything that I tell you now is going to answer that question right there. Describe in detail cellular respiration from the beginning to all of the end products. So it's 14 points, and I'm going to get a t-shirt that says, I don't make biology, I just teach it. Okay? So just know I'm doing my best to help teach you. I don't make it. So, all right. Now, things that we need to know. Who cares about cellular respiration? So the whole thing about cellular, I know, right? You care. You need energy. You need ATP energy. And so your plants undergo cellular respiration in their mitochondria. Your animals undergo cellular respiration in their mitochondria. Both plants and animals have mitochondria, okay? So let's go ahead and start out by making ourselves some nice little cells. I'll make a nice little big cell. We're gonna label structures first. Sometimes if you just label, the whole battle is in understanding what's taking place where. So there's my cell. Nice big cell, just like such. I think I need a better black. All right. So here you got this right here is a cytoplasm, cytoplasm. In the cytoplasm, you have a system called glycolysis. So glycolysis. Boom, we're gonna write all those goodies up there too. All right, so it's not gonna be to scale, it's gonna look really crazy, but I'm gonna draw a mitochondria. Mitochondria, I'm gonna make it really big because there's a lot of things going on in the mitochondria, so I just gotta make it really, really big. Now, when you give me the answer to the question, you can draw a picture, that's great. But you also have to use your words via sentences, just like I'm using my words and narrating as I'm drawing. Does that make sense? So pictures are nice, but they're not required. If you don't want to draw a picture, don't draw a picture. You can just give me straight up paragraphs to answer this question, and that would be fine. All right, so here's this brown is mitochondria, so I'm going to go ahead and label it here. M-I-T-O, chondrion. I'll put mitochondria. Mitochondria is plural. Mitochondrion is singular, but I'm just going to go ahead and do that. Your mitochondria is double membrane. It's got an outer membrane and it's got an inner membrane. Your inner membrane, inner folds, these are cristae. Now I'm drawing the cristae really small until I get to the end. I'm going to draw it really obviously stupid big because I got some work to do on it. Draw all throughout. And I always draw it too small. I'm just going to be honest with you. I really need to draw a lot bigger. Got a lot of work going on in there. The inner foldings of the inner membrane. So this is the inner membrane, and these inner folds are called cristae. So I will go ahead, I'm going to label cristae over here because I need that room to work. C-R-I-S-T-A-E. Now, inside the mitochondria, you have a semi-fluid liquid. It is called matrix. The matrix is just equivalent to cytoplasm, right? So let's go ahead and label it matrix. I'll go ahead and put it right here, mitochondrial matrix. And then you have your cristae. So there's your nice little structures. So cellular respiration is going to really officially be in 3 stages, but I'm going to break it down into 4 parts, okay? Officially in your book, 2 of the stages they meld together. They don't take it away, they just meld it together. But I'm going to go ahead and part it out, that way you can see, because they're taking place in very specific parts of the cell. So we have first stage, number 1, glycolysis. Glycolysis. And glycolysis takes place in the cytoplasm. Number 2, second stage is called pyruvate oxidation. Pyruvate oxidation. Pyruvate oxidation takes place in the matrix of the mitochondria. third stage is citric acid cycle. Which also takes place in matrix. Citric acid cycle is also known as the Krebs cycle. Citric acid is referring to a molecule that is in there. Krebs cycle is referring to the person who discovered it. And there's also triacetyl something cycle. You don't really see it being referred to that too often, but sometimes you do. So citric acid cycle, CYCLE. And then you have number 4. Fourth part is called oxidative phosphorylation. Oxidative phosphorylation. An oxidative phosphorylation is going to be split up into 2 parts. First part is called the electron transport chain. And then the finale of that is called chemiosmosis. So the oxidative phosphorylation, i.e. electron transport chain, and chemiosmosis, they all take place on the cristae. So you got your citric acid cycle, oxidative. I'm going to go ahead and write it on this cristae because I need this big cristae to work. So oxidative phosphorylation. I'm just going to draw that out. Just know it includes electron transport chain and chemiosmosis. So your biggest challenge at this point, figure out what your cell looks like. Figure out where each of the systems are taking place. That's step 1. You've already told me a bunch of stuff, so that's good. All right, so the whole point of cellular respiration is to produce usable energy called ATP. And ATP is going to be used for cellular work, right? So this is where it takes place. Let's go ahead and get started. I'm going to have some inputs and I'm going to have some outputs. My inputs, I'm going to go ahead and do with purple. My outputs, I'm going to do with red. All right. So let's start with the first system, glycolysis. Glycolysis takes place in the cytoplasm. Before you can get any energy out of any of this, you have to input sugar. You got to input a glucose molecule. So let's go ahead and do C6H12O6. I'll draw my little shape there, and I'll just let you know that that's glucose. So glucose is input. You eat your sugar. You get this molecule from your food you acquire, okay? Now, you input the energy. If you recall, glucose is a high energy molecule. The reason why it's a high energy molecule is because of the electron attractions, those covalent bonds that take place between all of the carbons in the glucose molecule. There's a lot of stored energy in there. And you haven't really seen it per se, I've mentioned it, but the stored energy comes from the sun via photosynthesis. So you ate your sugar that you got from your plants, and the plants photosynthesized it, took that solar energy, packaged it tight into this high potential energy molecule, okay? So you ate your glucose. Glucose is going to make way into your cells. In the cytoplasm, glycolysis is going to begin extracting energy from that glucose molecule. All right, so in order to do that, first what we need to do is we need to go ahead and input some energy to get it started. I need to move that cytoplasm. It's in my way. I got it right there. I'm going to write cytoplasm down here. CYTOPLASM. All right. So just like when you run out of gas, in your car, you're looking, and I don't know about you, I'm over here, I'm like, 5 miles till empty, 3 miles till empty, 2 miles till empty, right? Sometimes you're at 0. And it's like, you just got to get to the gas station. But if you didn't have actual gas in your tank, you can't get to the gas station to go get more energy, can you? So you have to have a little bit of energy before you can get more energy, okay? So in order to extract energy from this molecule, we got to input some of our cellular energy that's already sitting around, okay? And that's going to be ATP. You got to put in 2 ATP molecules. Now, if you've already gone through your glycolysis, you will look at your PowerPoints and you will see step by step by step, puts in an ATP, molecule change, puts in another ATP, molecule change. If you've already gone through it, you know what I'm talking about. If you haven't, you'll see. I'm giving you the overall big picture, okay? I'm not showing you step by step because then you'd really hate me. So I'm giving you the overall big picture. So you have to put into ATP. ATP is cellular energy, high energy molecule. It is going to basically help to run glycolysis. That way we can start oxidizing this molecule. And what oxidizing means, we'll hear it again, to oxidize, electrons are going to get removed from this molecule. You remember when I told you electrons, there's electron high energy in here that is making covalent bonds? Well, if I remove high energy electrons from that glucose, energy's coming with it. So just know your energy is going to follow your electrons, okay? All right, ATP is going to start running glycolysis, begin the oxidation, break down electron removals from the glucose. So when that happens, those electrons have to go somewhere. There's another set of molecules, they're called NAD+, nicotinamide, and there's 2 of them. So 2-nicotinamide, I'm gonna draw it like this. Oh, not very well, there we go. NAD+, you don't have to know nicotinamide, you can just know NAD+. And you do have to know NAD+, okay? The electrons from the glucose are going to get, not all of them immediately, a few of them, actually 4 of them, are going to get removed from that and given to this guy. Look at this, perfect puzzle piece shape to receive electrons. So your NAD+ are going to go into glycolysis, receive electrons, and it will come out, terrible red. I got a lot of red options here, let's see. Oh, that's a good one. They will come out as 2NADH. Now, there are hydrogens floating around here. Some of the hydrogens come from the glucose. I'm not going to make you be specific on how many hydrogens and where they're coming from, okay? I am labeling some things out. And so 2NADH is H molecules. The reason why they're NADHs is because the high energized electrons, came from here. And those high energized electrons are going to then basically covalently bond this proton with some hydrogen protons to make NADH. Okay, so this guy here, NAD+, when he goes in, receives electrons. He's an electron stealer. So his self-esteem is going to be reduced. So whenever you remove electrons, you steal electrons, that's reduction. This guy loses electrons, he's being oxidized. This guy gains electrons, he's being reduced. Okay, we'll talk about that more. Just kind of want to apply those vocabulary from last week. All right, so there's that. So this guy, if you recall, since he has those electrons, and those electrons are high energy. Why are they high energy? Because they came from the glucose. Glucose is a high potential energy molecule. Why is he high energy? Because it was made high energy from the sunlight. Okay? So this is a high potential energy molecule. This high potential energy molecule is temporary and it's just going to hold electrons in high potential energy. So we need to make some ATP in this system. And so out of that, you're going to you're going to get an input of 4 ADPs plus 4 inorganic phosphates. 4 ADPs plus 4 Ps. They're going to go into glycolysis, and because of the energy that is taking place, the step-by-step removal of energy from the glucose, not only do you get this molecule made, but you will get these bonded together to make 4 ATPs. 4 ATP molecules. High energy molecule used for cellular work. So, you had to input 2 ATP to get it started. But in the end, you got 4 out of it. So if you had a lemonade stand, and you wanted to make $4 out of your lemonade stand, but you had to input $2 into it to even buy lemons, what is your net profit? $2. Very good. So you had to put in 2 ATP to get out 4 ATP, so you really have a 2 net ATP. And the process in which this is made is called substrate level phosphorylation. I'm not going to show you what that looks like here. Once we're done with all of this, I'll show you what it looks like. As far as on here, all you got to let me know is via substrate. What does that mean? It means there's enzymes involved, right? Substrate level phosphorylation. Phosphorylation. That means there's going to be enzymes involved and a substrate molecule in order to bond the ADP and the 4 Ps together to make 4 ATP. It's real simple. It's a scary set of words. Don't let it scare you. It's just a set of words. Okay? All right. So, last but not least, we got to get some final molecules, and then I'll go ahead and work you through again. Final molecule coming out of glycolysis is going to be pyruvates. So, coming out and going in here, hopefully, 2 pyruvates. Pyruvates. 2 pyruvate molecules. These are the final molecules that will result when glycolysis is finished. So I'm going to walk you through this with my beautiful markers. These markers are carbons. 1, 2, 3, 4, 5. We need one more, and 6. This is my 6 carbon molecule called the glucose. Right? 6 carbons. 6 carbon molecule, glucose. Glucose is going to go into glycolysis. When it goes into glycolysis, right away it's going to split like that. And then you're going to end up with 2 ATP going in to remove some electrons. Oh, they get given to him, right? So the NAD plus come in, remove electrons, they get to go and become an NADH molecule. You also have 4 ADPs plus 4 Ps coming in, and you will get out of it 4 ATPs. In the end, you will have 2 pyruvates. 1, 2. Pyruvate is a 3 carbon molecule. 1, 2, 3. You got 2 of them. 1, 2, 3. These 2 pyruvates are going to enter into the mitochondria as long as there's enough oxygen present in the cells. So, if there's enough oxygen present in the cells, then oxygen just basically diffuses in. Boop, boop, O2. Lots of oxygen. Diffuses. You're oxygen rich, so then oxygen will diffuse into the mitochondria, O2. You breathe that in. So if there's not enough oxygen getting to the cells, these pyruvates cannot enter into pyruvate oxidation. They will end up getting recycled around via fermentation. But I'm not going to go over fermentation in this talk. Okay? Doesn't mean you don't need to know it. Just means it's not a part of this answer. All right. Why don't you go ahead and pause.